Okay, flowers, fruits, and seeds. We're moving past the plants, stems, roots, and leaves. And these are reproductive structures of flowering plants. So these are not applicable to all types of plants. Now, the plants have different lifespans, and we've mentioned this before. A plant cycles from seed germination to the mature plant, which can produce seeds. And you have a few different strategies, one of them being annuals. And these are mostly herbaceous plants. These are, well, all herbaceous plants. And they complete the whole cycle in one season. Then you also have another set of plants that do it in 2 seasons. So you have here an example of a biennial where it takes 2 seasons to fully make its seeds. And then you have perennials, which take multiple years in order to complete their full cycle, where they can make seeds. Okay, and we've mentioned this before and have shown a few differences between dicots and monocots, which is really a nickname which has continued even though we have kind of renamed them into 2 different groups: Magnoliopsida and Liliopsida. Okay. And dicots have 2 cotyledons in their seeds, where monocots have 1. Dicots have flowering parts in multiples of 4 or 5, so looking at the petals is usually the easiest way to do that, where monocots have multiples of 3. Monocots have parallel veins. Dicots have a network of veins, usually branched. Dicots have a vascular cambium and cork cambium present, especially if they are perennials. That is not present in monocots. The vascular bundles in the stems of dicots are in a ring, whereas they are scattered in monocots. And then another difference is in pollen grains. The pollen grains have 3 apertures and 1 aperture in the monocots. Okay, so we're going to go through the structure of flowers. So they start as an embryonic primordium, which then develops into a bud. And then the bud then develops into the different parts of the flower. The part in which the flower sits on is called the peduncle, and that may split into other branches called a pedicel, depending on the type of flower. The pedicel or peduncle then reaches a swollen part underneath the floral parts, which is called the receptacle. And then you have these whorls, or multiple parts, which kind of are all the same, called sepals, petals, stamens, and the pistil. So we'll start with the sepals, which is the outermost whorl, and collectively they form what's called the calyx, and this protects the flower while it's in the bud. Okay, so here we have sepals. All these together make the calyx. The next whorl inside is the petals. Okay, we can see 3 petals here. There's probably a 4th was cut off. And this forms what's called the corolla. And if they are bright or showy or have stripes or lines on them, usually they are to attract pollinators, maybe birds or bees or other types of insects. If they are inconspicuous or missing corollas, that happens in many trees, weeds, grasses, and wind-pollinated plants that don't need to attract pollinators. The calyx and corolla together are called the perianth in monocot flowers. Okay, so some of the other whorls include stamens, which attach around the base of the pistil, and these are what contain pollen grains. The filament is the stalk, okay, and then the anther is the part which has the pollen in it. The pistil then is the reproductive structure which contains the ovule, and it has a stigma on the top, a style, which is the stalk, and then the ovary, which contains the ovule. The ovary, for the most part, develops into fruit. The carpel then is modified leaves with ovules on the margins. Okay, and fused carpels make a compound ovary. So a pistil can consist of 1 to several carpels. And we'll see this in class when we look at flowers. A superior ovary sits above the flower parts, the petals and leaves and stamens. An inferior ovary has the receptacle below the flower parts. The ovary also contains ovules, okay, which develop into seeds after they've been fertilized. So flowers can be produced in a single stalk or in each with their own peduncle, or they can have multiple groups on one peduncle, which is called an inflorescence. And these are different types of inflorescences: a panicle, a raceme, a quorum, a umbel, and it's all just dependent on the shape, a catkin or a head. Okay, and those are some examples of them there. Fruits then are matured ovary and its accessory parts, and it contains seeds. So here is tomato, which you may have thought was a vegetable, but is actually a fruit. And it develops from flower ovaries only found in our flowering plants. So parts of the fruit, you have the pericarp, which is the fleshy part, and it has 3 parts within it. So here's the pericarp, endocarp on the inside, mesocarp in the middle, and exocarp on the outside. There are lots of different types of fruits. Some of them are exclusively the ovary and the seeds. Some of them can include adjacent flower parts, such as the receptacle. They can be fleshy or dry. So here's some fleshy ones. Here's some dry ones. And they can be split or not split. And they can be derived from one or more ovaries. And we'll go over these in lab. Fruits often have a part in seed dispersal. Some of them are dispersed by wind. You can see these samaras, which are they spin as they drop, helps disperse the seed further away from the parent plant. Some of them can be small and lightweight, so that they can just be lifted up by the wind. They may even have appendages, which are carried by the wind as well. Still, some other even take advantage of animals to disperse them. And that's where the fleshy part comes into place, where an animal will eat them and then pass the seed through the digestive tract, usually away from the parent plant. Some of them may adhere to the fur or feathers of animals. Velcro was actually invented based on some seeds' ability to attach to clothes. Oils can also be used to attract ants, such as elastomes on bleeding hearts. The ants will be attracted to the oils and will eat them, and then they'll take the seed and it will be planted wherever the ant takes it. Water dispersal can also be utilized by seeds by containing a section of air so that it will float, can go downstream from where its parent plant is. So the structure of seeds. The ovules are what develop into seeds, okay, after they've been fertilized. The cotyledons are the food storage organs. Again, you can have 1 or 2 cotyledons, depending on whether it's monocot or dicot. And the embryo then contains the cotyledon and the plant to combine. So this is the embryo. Within the embryo, you have the plumule, which is the embryo shoot. You see the little leaves starting to develop there. And you can have an epicotyl, where the stem is above the cotyledon. And you can't see that here, but we'll see that in a second. Or you can have the hypocotyl, which is the part below the cotyledon attachment. You also have the radicle, which forms the root. Alright, so in epigeneous germination, what you have is the hypocotyl lengthens and becomes hooked shaped. So this is hypocotyl here, and then the cotyledon is pulled above ground as it's hooked, and the radicle forms into the root. In hypogeneous germination, the hypocotyl remains short and the cotyledons don't emerge above the surface, so they stay below. So germination then is the process of a seed growing into a plant. So it's beginning to use the nutrients in the cotyledon to grow. Some require a period of dormancy, usually in temperate regions, and they usually don't start growing until they have some sort of awakening mechanism. So it may be mechanical or physiological factors such as hormones in the seed coat or the fruit, or It may be broken down by mechanical abrasion, thawing and freezing, bacteria presence, or soaking rains. All these things will change depending on the season of the year, and so they usually correspond with the springtime. Scarification is artificially breaking dormancy. So here we have someone using a file to scratch, mechanically alter this seed so that it will germinate. You can also soak them in water, or usually, if you don't scarify them, just putting them and planting them in the soil will give them what they need to germinate. After ripening, the embryo is composed of only a few cells, and then the seeds will not germinate until the embryo develops. The embryo has to develop, and then it will germinate and start growing. It's favorable in environmental factors needed for germination. Okay, so a lot of seeds may go in all different areas, but will only start to germinate when it lands in the soil and the sunlight and water and all the conditions required for growth. So water and oxygen, light or the absence of light, a proper temperature range, all that's important for germination, so that the enzymes can begin to function after the water is implanted within the seed. All right.